Welcome to the deep dive. Today we're tackling, well, really the bedrock chapter for college genetics, DNA and chromosome structure.
That's right. It's foundational.
Yeah, we're going to extract all those key concepts, the big discoveries, the molecular details you absolutely need. Our goal is to take you from, the very first inklings about genetic material all the way to understanding how it's packed so incredibly tightly in eukaryotes.
It really is an amazing story.
It is. And at its heart, it's kind of an architectural puzzle, isn't it? How do you figure out first that heredity is in the nucleus, then pinpoint the molecule, then understand its structure, and finally grasp the sheer engineering needed to, well, cram 6 feet of DNA into a microscopic cell nucleus?
And looking back, that journey wasn't straightforward at all. There was this major paradox early on.
Right, the protein versus nucleic acid
Yeah,
debate.
exactly. For a long time, the smart money was on proteins. They're complex, right? 20 different amino acids. It seemed obvious they had to carry the complex code of life.
Makes sense.
Well, nucleic acid seemed, frankly, too simple, chemically repetitive. They were sort of dismissed as maybe just structural scaffolding or something.
So, not the main player.
Not at all.
Hmm?
And overcoming that assumption, that bias, was the first big hurdle. It really sets the stage for the experiments that changed everything.
Okay, so that historical context is crucial. It makes those first discoveries even more significant. We should probably start back in 1868 with Johann Friedrich Miescher.
Ah, yes, Miescher, a young Swiss doctor.
Right, working with pus cells, basically, from discarded surgical bandages.
Not the most glamorous source material.
No, but he isolated this unusual substance from the nuclei. It was acidic and packed with nitrogen and phosphorus, which was odd, chemically speaking. He called it nuclein.
Even then, there was skepticism, right? He found something revolutionary, but getting it published was tough.
Yeah, he submitted the paper in 1869, but the journal editor apparently sat on it for 2 years.
2 years. Just couldn't believe something seemingly so simple could be important. It finally came out in 1871. Imagine that delay, the first glimpse of the hereditary substance held back, because it just didn't fit expectations.
Okay, so from Miescher's nuclein to the definitive proof, that takes us to bacteria and transformation.
Right. We have to talk about Streptococcus pneumonia. Griffith's experiments first showed this transforming principle. Something from dead virulent bacteria could make living non-virulent ones deadly.
But he didn't know what that principle was.
Exactly. That was the work of Avery, MacLeod, and McCarty in the 1940s. They nailed the molecular identity.
And their experiment was just elegant, wasn't it? Using enzymes as tools.
Beautifully designed. They made an extract from the heat-killed virulent cells, the stuff Griffith showed had the transforming principle. Then they treated portions of this extract with specific enzymes.
So one batch got RNase to destroy RNA.
Another got protease to destroy protein.
And the crucial one got DNase to destroy DNA.
Precisely. And the result? Only the DNase treatment stopped the extract from transforming the harmless bacteria into virulent ones.
So destroying the RNA didn't matter. Destroying the protein didn't matter.
Nope.
Only when they destroyed the DNA did the transformation stop. That had to be it.
It was incredibly strong evidence. DNA was the transforming principle.
But, seeing is believing sometimes. We needed something even more direct, maybe showing the genetic material actually getting injected.
And that's where Hershey and Chase come in, 1952, using bacteriophages viruses that infect bacteria, specifically T2 phage infecting E. coli.
This experiment is just brilliant in its simplicity. They used radioactivity.
Right, they exploited a basic chemical difference. DNA has phosphorus, but essentially no sulfur. Proteins have sulfur, but very little phosphorus.
So they could label them differentially.
Exactly. They grew one batch of phage with radioactive phosphorus, $3 labeling the DNA. Another batch with radioactive sulfur, $3 labeling the protein codes.
Then they let these labeled phages infect the E. coli.
And then came the clever bit, the kitchen blender.
Seriously, a blender.
Yep. They agitated the mixture in a blender. The sheer forces were enough to knock the empty phage protein coats off the outside of the bacterial cells without breaking the cells themselves.
Okay, so coats off, bacteria intact. Then they spun it down.
Right, centrifugation. The heavier bacterial cells formed a pellet at the bottom, and the lighter phage coats in liquid medium stayed in the supernatant above.
And the results were crystal clear.
Absolutely. When they used the 3 labeled phage, the DNA labeled the radioactivity was found in the pellet inside the bacterial cells.
Meaning the DNA went in.
And when they used the 3,5 labeled phage, the protein label, most of the radioactivity stayed in the supernatant with the detached coats.
The protein stayed outside, DNA carries the instructions, case closed for DNA and phages.
Pretty much nailed it for DNA as the general genetic material. But we do need to mention RNA's role too.
Right, because some viruses use RNA instead, like Tobacco Mosaic Virus, TMV.
Yes, and the proof for RNA came from Fraenkel-Conrat in 1957. Another elegant experiment involving reconstitution.
So they took different TMV strains apart.
Yeah, separated the RNA from the protein coats of two distinct strains. Then they mixed and matched, put the RNA from strain with the protein from strain B, and vice versa. Created hybrid viruses.
Okay, and then infected plants with these hybrids.
Exactly. And the key finding was the progeny viruses produced in the infected leaves were always identical to the strain that donated the RNA. Didn't matter which protein code it started with.
So the RNA dictated the outcome. It held the genetic information for those viruses.
Confirmed RNA's role as genetic material in that context.
Okay, so now we know what it is, DNA, or sometimes RNA. Let's get into the how, the structure, the double helix.
Right. The building blocks first. Nucleic acids are polymers, long chains made of nucleotides.
And each nucleotide has 3 parts, right?
3 parts. A phosphate group, a 5-carbon sugar, a pentose, and a nitrogen-containing base.
We need to be clear on the differences between DNA and RNA here. The sugar is key.
Absolutely. RNA uses ribose. DNA uses 2-deoxyribose. That deoxy means it's missing an oxygen atom, specifically the hydroxyl group at the 2' carbon position.
And that missing oxygen makes DNA more stable chemically.
Much more stable, yeah. Less reactive.
Yeah.
Then there are the bases. Both DNA and RNA use adenine A, guanine G, and cytosine C.
But the 4th base is different.
Right. DNA uses thymine, T.
Oh, yeah.
RNA uses uracil, U, instead. You have to remember that swap. T in DNA, U in RNA.
And we group the bases, too, by their structure. Purines and pyrimidines.
Correct. A and G are purines. They have a double ring structure.
Yeah.
C, T, and U are pyrimidine single ring structures. Remember, pyrimidines has gold, AG. Pyrimidines cut the pie, C, T, and U.
Okay, got the components. Now, the big picture. The double helix structure deduced by Watson and Crick in 1953, they built on some crucial earlier work.
Oh, definitely. 2 main lines of evidence were indispensable. First, Erwin Chargaff's chemical analysis.
Chargaff's rules.
Yes. He carefully measured the amounts of the bases in DNA from different organisms, and he consistently found that the amount of adenine roughly equaled the amount of thymine, AGT, and the amount of guanine roughly equaled cytosine, GC.
Which strongly suggested pairing, right? A with T, G with C.
It screamed pairing. It wasn't proof of how they paired, but the fixed ratios were undeniable.
And the second piece, the X-ray diffraction images.
Crucial work by Rosalind Franklin and Maurice Wilkins. Their X-ray patterns showed DNA was a helix, highly ordered, with repeating structural features. That famous X pattern in Franklin's photo 51 indicated a helical structure.
And it gave dimensions, too, like the 0.34 nanometer spacing between the stacked units.
Exactly. Those dimensions were vital clues. Putting Chargaff's rules together with the X-ray data, that led Watson and Crick to their model.
The bDNA model, let's break that down.
Okay, it's a right-handed double helix. 2 polynucleotide strands coiled around a central axis. And critically, the strands are anti-parallel.
Meaning they run in opposite directions. One goes 5' to 3', the other goes 3' to 5'.
Correct. And the bases are on the inside, paired up very specifically according to Chargaff's rules. Adenine A always pairs with thymine T, using 2 hydrogen bonds.
And guanine G always pairs with cytosine C using 3 hydrogen bonds.
Right. This specific AT and GC pairing establishes complementarity. If you know the sequence of one strand, you automatically know the sequence of the other.
And that complementarity immediately suggests how DNA could be copied, right?
Absolutely. It was a huge insight. Now, what holds it together? The hydrogen bonds between the pairs are important for specificity.
But they're relatively weak individually.
They are. The real stability of the helix comes from hydrophobic stacking forces. The flat base pairs stack on top of each other, perpendicular to the helix axis, kind of like a stack of coins.
And these bases are hydrophobic, they don't like water, so stuffing them into the core of the helix away from the watery environment is energetically favorable.
Exactly. That stacking interaction is incredibly strong in aggregate and provides the main stabilizing force for the double helix. Water is excluded from the core.
The structure also has grooves, doesn't it?
Yes, because of the way that backbones twist, it creates 2 grooves on the surface. A wider major groove and a narrower minor groove.
And these aren't just incidental features.
Not at all. They're critical recognition sites. Proteins that need to read the DNA sequence, like transcription factors, often interact with the edges of the base pairs exposed in these grooves. Especially the major groove, which offers more information.
Now, bDNA, the Watson-Crick model, that's the main form in our cells.
It is, under physiological conditions. It has about 10 base pairs per helical turn. But there are other forms DNA can adopt.
Like aDNA and zDNA.
Right, aDNA is also right-handed, but it's shorter and thicker, about 11 base pairs per turn. You tend to see it when DNA is dehydrated, or in RNA-DNA hybrids, or double-stranded RNA.
And zDNA is the odd one out.
It really is. It's a left-handed helix, much thinner, with a zigzagging backbone, hence the Z. It has 12 base pairs per turn and tends to form in sequences with alternating purines and pyrimidines, especially Gs and Cs. Its biological role is still debated, maybe something in gene regulation.
Okay, regardless of the exact A, D, or Z form, there's another layer of structure crucial for function inside the cell. Supercoiling.
Yes, supercoiling is essential. Think it's twisting a rubber band until it coils up on itself. DNA does that too.
Why? Because the ends are fixed.
Exactly. In prokaryotes, the chromosome is circular. In eukaryotes, the linear chromosomes are organized into loops anchored to a protein scaffold. So the DNA can't freely rotate to relieve torsional stress.
And this leads to coiling of the coil.
Right. And the form that dominates in living cells is negative supercoiling. This means the DNA is slightly underwound relative to the relaxed B-form state.
Why underwound? What's the advantage?
That underwinding introduces strain, like storing energy in the molecule. This strain makes it easier to separate the two DNA strands locally.
Which you absolutely need to do for replication and transcription.
Precisely. Negative supercoiling primes the DNA, making those processes more accessible and efficient. It's a fundamental functional requirement.
Okay, we've got the molecular structure down. Now let's zoom out and tackle the packaging problem. This is where the scale becomes just mind-boggling.
It really is. You mentioned the mouse chromosome example earlier. Stretching out the DNA from just one chromosome gives you 60 millimeters.
6 centimeters. And it has to fit inside a nucleus maybe a few micrometers across. And during cell division, condense into a visible chromosome maybe 10 micrometers long.
That's a compaction ratio of several thousand-fold lengthwise. It's an incredible feat of biological engineering.
How was it achieved? Let's start with prokaryotes, which are simpler.
Right. E. coli, for instance. Its DNA is about 1,500 micrometers long, that's 1.5 millimeters, but the cell itself is only 1 or 2 micrometers long.
Still needs packing, just not as extreme.
Correct. The E. coli chromosome exists as a folded genome. It's organized into roughly 50 to 100 distinct loops or domains radiating from a central core.
And each loop is independently supercoiled.
Yes, independently and negatively supercoiled, which helps compact it. This structure seems to be stabilized by both proteins and RNA molecules. If you treat the cells with RNAs, the folded genome partially unfolds.
Okay, so loops and supercoiling in bacteria, eukaryotes must be way more complex.
Oh, vastly more complex. Eukaryotic chromosomes are a composite material called chromatin. It's DNA complex with a whole suite of proteins.
2 main types of proteins, right? Histones and non-histones.
Exactly. The histones are the main structural proteins. They're small, rich in basic amino acids like lysine and arginine, giving them a positive charge. This lets them bind tightly to the negatively charged phosphate backbone of DNA.
And they're highly conserved across eukaryotes.
Remarkably conserved. There are 5 main types: H1, H2A, H2B, H3, and H4. H2A, H2B, H3, and H4 form the core, while H1 is a linker histone.
Then there are the non-histone chromosomal proteins.
Yes, a much more diverse group. They're typically acidic, negatively charged, and vary greatly between cell types and organisms. They include enzymes involved in DNA replication and repair, transcription factors, scaffold proteins, lots of regulatory and structural roles.
So how do these components work together to achieve that massive compaction? It happens in stages.
Yes, 3 main levels of folding are generally recognized. Level 1 is the formation of nucleosomes. This is the fundamental repeating unit of chromatin.
The beads on a string structure you see in electron micrographs.
That's the one. The bead is the nucleosome core. It consists of 146 base pairs of DNA wrapped about 1.65 times around an octamer of histones, 2 copies each of H2A, H2B, H3, and H4.
Okay, so the core particle is DNA wrapped around 8 histone proteins.
Right. Then the string is the linker DNA connecting adjacent nucleosomes. And the complete nucleosome includes the core particle plus 1 molecule of histone H1, which binds to the linker DNA where it enters and exits the core, kind of like a clamp.
And this first level already achieves some compaction.
It does. It shortens the DNA length about 7-fold and creates that 11-nanometer diameter fiber.
Okay, level 1, nucleosomes. What's level 2?
Level 2 involves coiling or folding that 11-nanometer fiber into a thicker structure, the 30-nanometer chromatin fiber. How exactly this happens is still debated. There's the solenoid model where the nucleosomes coil helically and the zigzag model where they fold back and forth.
But either way, it gets thicker and more compact.
Exactly. And histone H1 is absolutely crucial for forming and stabilizing this 30 nanometer fiber. This level achieves another, say, 6 to 7-fold compaction.
So we're maybe around 40 or 50-fold compaction total now. Still a long way to go for metaphase chromosomes. What's level 3?
Level 3 is the organization of that 30 nanometer fiber into large loops. These loops, containing tens or hundreds of thousands of base pairs, are thought to be anchored at their bases to a central protein scaffold.
A scaffold made of non-histone proteins.
Yes, entirely non-histone proteins form this dynamic structural core. These loops get further coiled and folded, ultimately achieving the maximal condensation we see in a metaphase chromosome, that final several thousand-fold compaction.
Wow. Okay, so nucleosomes, 30 nanometer fiber, and scaffold loops. That's the hierarchy. Before we wrap up, we should touch on some special features of eukaryotic chromosomes, especially related to all that non-coding DNA.
Right, the sheer amount of DNA in eukaryotes that doesn't code for proteins is staggering. Humans have maybe 700 times more DNA than E. coli, but only about 4.5 times more genes. A lot of the rest is repetitive DNA.
It used to be called junk DNA, but we know better now. Much of it is structurally vital.
Absolutely. And we could actually detect how repetitive sequences are using a technique called renaturation kinetics.
How does that work?
Well, you take DNA, heat it to separate the strands, denature it, and then let it cool slowly so the complementary strands can find each other and reform double helices, renature or anneal. Highly repetitive sequences renature much faster than unique sequences. Why? Because there are many copies, so a matching partner is easier to find just by random collision. It's a concentration effect.
Like finding a friend in a small village versus a huge city.
Exactly. And this technique revealed that a large fraction of eukaryotic DNA consists of these rapidly reannealing repetitive sequences, often clustered in specific regions.
Like centromeres and telomeres.
Precisely. Let's define those. The centromere is that constricted region you see on a metaphase chromosome. Its key function is to serve as the attachment site for spindle microtubules during cell division.
Via the kinetochore complex that assembles there.
Right. It ensures that sister chromatids get pulled to opposite poles correctly. Structurally, centromeres are embedded in heterochromatin, highly condensed chromatin, and typically contain massive arrays of repetitive DNA. In humans, it's the alpha satellite sequence, repeated over and over for hundreds of thousands, even millions of base pairs.
Essential for chromosome segregation. What about telomeres?
Telomeres are the natural ends of linear eukaryotic chromosomes. They're absolutely critical.
Why? What do they do?
They have several jobs. First, they act like protective caps. They prevent the chromosome ends from being mistaken for broken DNA that needs repair, and they stop chromosomes from fusing together end to end. They also resist degradation by nucleuses. And, importantly, they play a role in ensuring the very ends of the chromosome can be fully replicated.
And structurally. Also repetitive DNA.
Yes, telomeres consist of short, tandemly repeated sequences. The sequence is highly conserved across vertebrates in humans. It's TTGG, repeated hundreds or thousands of times.
And there's a special structure at the very end.
There is. The strand running 5 to 3/4 towards the end is G-rich, and it extends beyond the C-rich complementary strand, creating a 3-foot single-stranded overhang. This overhang is key to the protective mechanism.
How does it protect the end?
It actually loops back and invades the upstream double-stranded region of the telomere repeats, displacing one strand and pairing with the other. This forms a lariat-like structure called a T-loop.
So it tucks the very end away into a loop.
Exactly. It hides the vulnerable free DNA end. And this structure is then stabilized and shielded by a complex of specialized proteins called shelterin. Proteins like TRF1, TRF2, and POT1 bind to the telomeric DNA, especially the T-loop and the overhang, essentially putting a protective cap on the cap.
Fascinating. So the T-loop and shelterin complex work together to maintain telomere integrity.
They do. It's a sophisticated solution to the problem of having linear chromosome ends.
Okay, this brings us pretty much full circle then. We've gone from Miescher isolating nuclein without knowing its significance.
Through the definitive experiments, proving DNA, and sometimes RNA, holds the genetic code.
To the elegant Watson-Crick double helix model explaining complementarity and stability.
And finally, to the incredible multi-level packaging using histones and scaffold proteins to condense meters of DNA into microscopic chromosomes, complete with specialized centromeres and telomeres.
Yeah, the sheer scale is just astounding. That 60-millimeter mouse chromosome DNA packed down into a 10-micrometer metaphase structure.
That condensation, that packaging, enabled by histones and non-histone proteins, is just as crucial for eukaryotic life as the DNA sequence itself. You can't separate the code from its physical management.
So let's leave our listeners with a final thought to ponder, building on all this. Considering the huge differences simple circular DNA in prokaryotes versus these massive linear, highly packaged chromosomes in eukaryotes, what are some of the fundamental challenges eukaryotes face in processes like DNA replication and repair that prokaryotes just don't have to deal with? Think about why telomeres and centromeres aren't just useful, but absolutely essential for eukaryotes.
That's a great question to reflect on. The solutions, telomeres, centromeres, complex repair pathways, really highlight the consequences of eukaryotic genome architecture.
Indeed. Well, thank you for walking us through that complex material, and thank you, our listener, for providing the source material that fueled this deep dive into genetics fundamentals. We'll catch you on the next one.